Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. Excellent. Well, delighted, Jim and Pam, that you are joining us today. We're super excited about this conference in February. It's coming up. It's amazing how quickly time passes. Perhaps we can start with, maybe it's a trite question, but I think people like to know, to get a little bit of sense for how you both, Jim and Pam, found yourselves fascinated and very much wanting to develop the cancer field. What brought you to MD Anderson, and just, you both have such historical, phenomenal successes, but give us a little sense for some of the motivations that led you to MD Anderson.
Sure. I guess I'll start and just say that, cancer affects so many of us and our families. And my grandmother passed away with pancreatic cancer. So I always had an interest in, entering into oncology as part of my medical career. I did a PhD in immunology, and I always thought it was fascinating how the immune system is so powerful to reject virus or bacteria or infectious organisms in our body. And thought, yeah, people were always talking about about the idea that the immune system can also reject tumors. And so we were doing that in mouse models in my studies in the lab. So I really wanted to see how we can bring this to patients. And I joined MD Anderson after finishing my postdoctoral fellowship in Lloyd Oles' lab, where he was working on vaccines for cancer patients. And then when I joined MD Anderson, I started working on immune checkpoint therapies for cancer patients, and participated in and wrote and many of the studies that led to FDA approvals for the immune checkpoint therapy agents that Jim developed.
Phenomenal work. And Jim, a little bit about the history that's led you here, abbreviated version.
Well, I also, my family suffered considerably from cancer. My mother died when I was young, and both of her brothers died of lymphoma. Both of her brothers died of cancer. I lost a brother to cancer, and have another one who had cancer. And all this, seeing a lot of the impacts of therapy and everything made me think that we could do better. I started college and learned about the immune system just after T cells were developed, and was told by a professor, he wasn't certain they existed, but if they did, there were these things as proposed that went through the body and eliminated, virus infected cells, maybe even tumor cells, and obviously that seemed to me to be a really clean way of going about it. So beginning in the early 1980s, I started working on what regulates T cells, turns them on, and lucky enough discovered also a break we called CTLA4 that had we showed in the laboratory blocking that could lead to rejection of essentially freed T cells to attack almost any kind of cancer. And moved from Berkeley, where a lot of that work was done, to Sloan Kettering to be there while the phase 3 trials were going on, participate in that. After approval, I decided to shift to more mechanistic studies and trying to understand how it worked. And Dr. Sharma had just moved here and told me about some of the things she was doing, and really working out ways of getting patient material and dissecting them carefully, bringing what we were doing in mice, bringing that to patients. And so it seemed like a good place to come and team up with her, take begin to provide, as I said, mechanistic studies to really try to develop an understanding of how immuno-oncology drugs work, why they don't work, they don't, and how to manage them properly. So,
It's I think it's just extraordinarily inspiring to hear how the people, your families, really sort of drove, I think, the ambition to address cancer and its effect on people we love and the people we live with, and also the greater community at large. And I think since maybe you mentioned CTLA-4, and I think since that 96 paper in Science where you
Yeah.
show that, blocking CTLA-4 can enhance anti-tumor activity, a lot has happened, and I think an enormous field has emerged from these fundamental insights that you've achieved. And I guess there are so many questions, but perhaps, excuse me, the first question I might pose to both of you, because I think this is something you both work on and deeply care about, is that with the initial successes of this immune checkpoint therapy, one of the challenges is that it doesn't necessarily work for everybody all the time.
Mhm.
And so you have responders, excuse me, non-responders. And so could you speak to what it is that you think accounts, at least in part, for how people differentially respond or do not respond as individuals? Your families perhaps succumb to different types of cancers, and just sort of how you're approaching that heterogeneity in terms of response to immunotherapy.
Yeah, I think, when you look at mouse models, Jim did so many mouse models where the immune checkpoint therapies worked beautifully in the mouse models. And then when we translated this into the clinic, we wrote the first clinical trial as a pre-surgical or neoadjuvant trial looking in bladder cancer patients to try and understand what exactly are the immune responses in the tumor tissues that lead to these responses or the non-response. And we were able to really understand at that time that the human immune response is going to be different, obviously, right? It's not the same as the mouse immune response. I mean, when mice were injecting the tumor cells into the mice and then treating them within a few days or a few weeks, whereas in humans, they've had these cancers for long periods of time, and the immune system has evolved over time with them. The other thing that we understood was that Jim's work was about targeting T cells and how T cell responses evolve. But the immune system is made up of so many different cell types: B cells, dendritic cells, myeloid cells, NK cells, and all of these different cells have to play a role and communicate in different ways, not only in driving the response to lead to tumor rejection, but also suppressing the response so that we don't get ongoing immune responses that can lead to autoimmunity. So, understanding the mechanisms that drive a response to get tumor rejection, but also control the response was very important because we have to understand how the different cells interact with each other, what kind of neighborhoods and communications they have. And we can't just focus on the T cells alone. So I think these are the things that we're trying to understand. Some tumors, like pancreatic cancer or glioblastoma, they have a lot of myeloid cells in them. They don't actually have a lot of T cells. So they have some, but not a lot, where the myeloid cells tend to be very immunosuppressive. They also have other cells, the cancer-associated fibroblasts, that we're still trying to understand how to target. And now you're going to hear in this conference about the nerves, right? Because the nerves also impact the immune response, and the nerve innervation of these tumors are definitely impacting the immune response. You'll also hear about microbiome from other people, right? Your gut microbiome or microbiome in the tumors themselves, how that can impact immune response. I think the purpose of launching the James P. Allison Institute was to take the expertise from everyone in these different fields: neuroscience, microbiome, protein folding and design for drug development, myeloid cells, B cells, T cells. Take all of these different expertise under one institution that can then take the data from all of these different groups to combine efforts in improving immune checkpoint therapy with new combinations for the different tumor types. So I think that is what we're working on now. And I'm really hopeful and optimistic based on, first of all, the brilliant people that are working with us. Um, and the encouraging data that we're seeing from some of the new combinations.
Hmm. I mean, a lot of heterogeneity in the cell types that are engaged in this tumor response, and also I'm guessing a lot of heterogeneity in the life history of the people that you're treating. But
Yeah.
please, Jim, your thoughts?
Yeah, I would add one of the things that we're learning with the application of spatial transcriptomics, and as Dr. Sharma is mentioning, is that there are these cell types that are there, but if you get to the point where you can look at them individually in the tissue, you see that different neighborhoods of what appear to be the same cells can be quite different depending on where they are. For example, Linghua Wang from our institute has shown that fibroblasts even can acquire an immunosuppressive function if they're growing in an area near where there's their T cells and things going on. So it's even the cells that we thought before were inert or not, and I think this probably includes nerve cells as well. And there are a lot of circuits that we don't understand that we're still beginning to put together. And as we dig deeper and deeper with these new technologies that are becoming available, I think we're able to understand some of it. But I think that hopefully it's not going to be that every patient responds individually to different combination of drugs, but there may be some patterns depending on the disease and where they started and all this that we hope will emerge from the studies that we're all involved in. But I think I'd like to point out that while not everybody responds, there are about 300 to 500,000, depending on how you count it, of patients that are currently living with durable remissions from one sort of a cancer or another. That's a big number, and it's growing every day. The percentage of patients that are because of new approvals, are able to get treatment, it's approaching between 40 and 50% now of all cancer patients. So we've got a lot to learn, a lot to do, but it's a field that's not quite reached its peak yet. We've got a lot more to understand. And so it's great to have this opportunity to put what we understand with Moran's work and others showing the interaction between different cell types that we never dreamed of before, but now we can begin to start to put them together. Again, as I said, with new spatial transcriptomics and proteomics, we could hopefully get down to narrowing down what individual cells are doing in a tumor. So.
Yeah, I think that's fascinating. I think these technologies like spatial transcriptomics and proteomics speak to this possibility that you mentioned of patterns in response, and perhaps even predictive patterns. And so perhaps you and Dr. Sharma could speak to this idea of predictive biomarkers, whether those are biomarkers that are circulating, that could maybe stratify people in terms of treatment likelihood of outcome versus severities, and not just circulated biomarkers, but maybe perhaps even more complicated landscape biomarkers looking at transcriptomics. So how can we predict how people are going to do in response to these phenomenal therapies that you're developing?
Yeah, I think biomarkers are so important for helping us to select the right patients for the right treatment. And I do think that there are some biomarkers when we combine the genomic medicine space with the immunology space, we're starting to see that certain mutations, for example, predict for better outcomes, right? That is why we have the microsatellite instability high mutation as a FDA approval, right? Which is unheard of previously to have had something like that as an FDA approval, which is across many different tumor types. And I think that's the venue in which we're working now, is looking at these biomarkers from combination standpoint of the biomarkers themselves, combining the genomic medicine field with immunology, combining maybe cancer neuroscience with immunology, right? So that we're taking advantage of the whole microenvironment, that we understand what's happening in the tumor cells, what's happening in the immune cells, what's happening in the nerves, what's happening, in the fibroblasts. Maybe there's a way of combining these different pieces. I will say, for example, we recently showed that clonal hematopoiesis, which, as you know, is a condition that affects many, many different individuals. We, especially as you get older, we found that the TET2 mutation of clonal hematopoiesis in TET2 really predicts for better outcomes with immune checkpoint therapy, even though in other groups they've shown that by itself, obviously, it can drive leukemias, for example, and those kinds of things, or be bad for outcomes in cancer. But in the setting of immune checkpoint therapy, it can definitely have a better outcome. And there are ways to think about this, even, for example, surgery. If you have stage 4 cancer throughout your body, you're not going to do surgery with stage 4 cancer, because if you remove one lesion, the other lesions will grow. And we've tried to say, well, maybe we could do chemotherapy and surgery in stage 4 setting, and targeted therapies, for example, take TKI inhibitors. And those data have shown that you can't combine with surgery and get better outcomes. What we think is that in the setting of immunotherapy, maybe you can combine surgery to remove one lesion and immunotherapy to treat the rest, because the removing of one lesion is an antigen release that allows them to prime the immune response. So even in that setting now, we're beginning to show that there are things that we can do to predict better outcomes. So if a patient can go to surgery for a single lesion, maybe that patient will have a better outcome with immune checkpoint therapy. So where's the field of biomarkers going? Could be multiple different areas when you think of biomarkers. They're not just a standard, biomarkers from blood-based biomarkers that we think about. Of course, we want to get the blood-based biomarkers. I think like circulating tumor DNA and these kinds of things would also be helpful as we start to incorporate them. But I do think we're broadening our thought process of how we think of biomarkers and what we're going to use for selecting the right patients for some of these combination strategies like surgery plus immunotherapy or immune checkpoint therapy for clonal hematopoiesis mutations of TET2.
I was just going to say it may take on treatment biopsies early on to do sort of to get hints from the things that Dr. Sharma was talking about, because it baseline when you start giving these drugs, everything changes. If you take one checkpoint off, the immune system responds by putting other ones up, and there may be patterns to to those sorts of things that emerge, but we won't know it until we do on treatment biopsies. Maybe there'll be some way to link, as was discussed, the genetic and the baseline status of the tumor, but I think more likely it's going to require on treatment biopsy and then subsequent decisions made from that point based on.
I wanted to maybe drift a little bit from like the hardcore science. I have a question that I've been thinking about for a long time, and I think Jim, you might be the right person to answer this. Like there are a lot of parallelities between the field of cancer neuroscience today to, I guess, immunology 60, 70 years ago, and I want to take you for your time in Berkeley, when you started realizing that something is happening, like, was this like a process that you realized that you're onto something that's going to change the way we treat patients, or it was like a wow moment, like a eureka moment? Because in the field of cancer neuroscience today, I experience this almost every day. You discover something new every day, like, oh, this nerve is actually doing that, and this nerve exists in this tumor, and I can't believe they're interacting. And you start getting the feeling that there's something there. And I wanted to hear about your experience around that time. What exactly was the process for you, or was it like a moment?
Well, basically, what we're doing is just trying to figure out what turned T cells on and off, and when we began this work, there was a question of whether there were even antigens, really on tumor cells. But we showed that almost any tumor, if you provide a second signal that people didn't appreciate, the co-stimulatory signal to them, the antigens were there, and you could get antigen recognition. It's just that tumors don't give all the signals, and so we found out a way to keep them from getting the off signal eventually that they get that tells them not to respond anymore. But I didn't really think that it would quite do what it's done. I thought it would help with some cancers, but it turns out it's a pretty successful beginning to learn how to treat some cancers. Now it opened, as I'm sure this new field that you're helping open, it's going to reveal all kinds of new molecular targets down the road that we can address to use for diagnostics, prognostics, and for therapeutic targets, I think. So it's a process. You learn every day.
Well, Moran, I think it's so exciting that you are finding new things and you're so excited by it, because I do think that's the passion that drives the field, right? I mean, in a lot of ways, we were so used as immunologists, especially immunologists who wanted to treat cancer, right, with the immune system, was sort of like a joke and people didn't take it seriously. And we were like, the last day of the meeting in the last session where nobody attended, trying to present our new data and findings. I mean, now you can't even get into the room when there's an immunotherapy talk. But back then there were like 10 or 20 of us that was always at each other's talks, and it was just us as a group trying to be supportive of each other's data and findings. And I think that's where you are right now in the cancer neuroscience space. And all I would say is that, it takes one person to sort of have that drive and passion and that persistence. And, if anything, Jim is the most persistent person I know when it comes to scientific data, and you let the data speak for itself. You shout it as much as you can, but eventually people will start to hear about it, and you will see the field open up more. And especially now, if you can figure out how to apply that clinically to treat patients because of the data that you're generating, then the field, of course, explodes in a whole other format. So I would just say that, I think we're all grateful for the fact that there are persistent people who, keep pushing us forward in new directions and thinking out of the box. And so I certainly applaud you in that effort.
Absolutely. I think we all love Moran for this, and
Yeah.
it speaks to I think this deeper human element of human compassion driving scientific inquiry,
Yeah.
and recognizing that behind a lot of science are people who care very deeply about the human condition. And this is really what is prompting their exploration of the rules of the game. I wanted to come back to the data capture and the technology and the biomarkers and the heterogeneity that I think we're speaking to, both in terms of the people and their histories prior to cancer, and in the process of managing the cancer. Um, Surely machine learning and AI are going to play a role in analyzing these large data sets. Could you speak to the benefits and also the risks of using some of these machine learning languages and AI tools that are some have argued are limited by the kinds of information that they use as their training sets. So I'm sure it's a tool that can be used and can be beneficial, but sort of speak to the benefits and the potential risks.
People say all the time, garbage in, garbage out, right? So we have to be careful that we're not putting in a lot of things into the machine learning system whereby they can't really have all of the correct information to make analyses or predict what the outcome should be. So we have to be thoughtful about that. And I think a lot of times what we have in our data systems where we're capturing clinical data, patient heterogeneity, their background, what they ate for breakfast, what medications they're on, all those different things have to be compiled in a way that you can load it into an appropriate AI system. And so I think the medical records system are a little bit lagging behind in terms of how things are captured. And then how do those medical records all speak together, right? I mean, the medical records maybe at one institution is captured in a different way than the medical records at a different institution. And as you know, AI systems need thousands, if not millions and millions of records before they can start to analyze the data and come up with patterns. And so we have to think about how we put patient information into these systems, because again, we want to be protective of our patient privacy and their medical information, but we also want to share information across systems so that the AI tools can have a large enough data sets with accurate information to start to make these connections about patterns that can be used to really help our patients. So I think it's going to have to be a partnership. There'll have to be a partnership with academic institutions, the federal government playing a role in how this will go, what are the regulations, and of course, all of the big tech firms that are out there in Silicon Valley that have developed these incredible AI systems for, my goodness, I could type the word shoe into my browser and every shoe on the planet will show up, so I want to be able to do that for cancer. And, I want to be able to do what we do all the time with our Google browser or any other browser that we use, to be able to put things in there and have accurate information show up for us. And in this age of misinformation and disinformation, we have to be very careful that we're not providing advice to patients through an AI system that's telling them things that could actually hurt them. And that would be my concern. I can't tell you how many times just in the last week I've had patients come in who are on ivermectin, and I'm very worried that ivermectin is not a treatment for cancer, and I want them to get the appropriate treatment for cancer rather than something that they may have, gotten in some sort of misinformed way.
And of course, there's the scourge of Tylenol that we heard about in the last couple of days. But anyway, one of the things that I would like to say about AI things, I think that I agree with everything Pam said. The problem is, I think that large data sets, if we could figure out how to get spatial transcriptomics and proteomics in there, which is a problem right now, understanding how to even get that data along with the clinical data. But if we could do that, begin to see patterns emerge, it's going to hopefully generate hypotheses. I don't think that the AI systems are going to give us answers. They're going to give us possibilities, and we're going to have to go in then and generate hypotheses and test and make sure that what's suggested is correct, and bring and just bring the creative mind back to understanding what's important and what features might be less important, and decide where to take the data.
In that sense, I tend to agree. Like if you tend to think about this as a tool more than just a resource, and you master how to use this, then you will use it in a healthy way, and it goes a long way. In terms of also education, how we educate our students. I'm in the graduate school, and it's challenging, because they can use this for almost every task that you give them. So how do you actually trigger them to use their neurons in order to start developing new concept, new hypothesis, like Jim mentioned. I wanted to ask you both, like again, the field of cancer neuroscience is bubbling. There's a lot of new ideas, a lot of people there. What advice will you give us as scientists who want to translate this into the clinical realm, um, that will help us, to like go through the barrier, or diffuse those barriers, and like you said in your great feature, Jim, like get it to my patients as quickly as possible.
Yeah. Well, I think one thing is to not rely back to something Dr. Sharma said, is you could go only so far with mice. And rather than trying to find the perfect mouse model and answer all the questions, if you've got a good general principle that seems sound and you can show it's safe, it should it should go into patients earlier rather than later, so that you don't spend five or like as I had to do 4 or 5 years just waiting for something to happen. You could at least rule something in or out, and we could do mechanistic studies now to know whether something's got a chance of working or not with very early in the therapeutic regimen. So I think that I just hope that we'll move things faster to the clinic than we have been.
Yeah, I think you can test your hypotheses in patients, right? I mean, if you have enough data in the mouse models and you can generate some safety data in some sort of phase 1 study, and then just, look for whether or not you can do a small 10 or 20 patient trial. That's what we did with immune checkpoint therapy with anti-CTLA-4 and bladder cancer patients before any immune checkpoint therapy agents were even FDA approved. So it was very early with these drugs, but we were testing them in patients to really understand the patient's immune response. And I think that can be done if you have some hypotheses that you can test in your patients to see if you're actually generating the response or having the outcomes that you predict based on the mechanisms that you understand.
So basically, what I'm hearing is, like if you have like good data in your preclinical model, and you don't really have to optimize it, and at that point, just be brave and just do like a small pilot feasibility safety trial to try and get, like the safety data that you need, and also the signal about the efficacy. And this is an important message that I want to relate to everyone who's listening, because um, we're very close to that point in the field of cancer neuroscience, and hearing this from you guys, like as an authoritative, individuals in the field, it's really important. Um, in the same line of thought, um, the way I think about the nervous and the immune system is that they're both evolved to protect the host in a very different fashion. The immune system is more adaptive and maybe a little bit slower to respond, but you do have the acute responders, or the first, and the nervous system is mostly sensing, but also executes some functions. That's one of the reasons why our forefathers or foremothers weren't eaten by cheetah. We sensed the cheetah and we ran away, right? So it's a protective mechanism, very different from T cells. But um, what are your thoughts about the crosstalk between those two? Like, because they're in the field of microbiology and autoimmune disease, it's well established. Even in asthma or atopic dermatitis, we know that all of those interactions occurring, but we don't know much on cancer and how it happens in cancer in that sense.
So you're asking about the crosstalk between nerves and the immune system specifically?
Correct. Yes.
Yeah, I think there's some really nice papers that were published. I think looking at TACMAX group, for example, and of course your own work, Moran, showing that clearly that neurotransmitters are playing a role in affecting immune responses. And so, understanding at which point those nerves are firing to, have neurotransmitters released and processed within the immune system. I think the mechanisms behind that are still the things that we're trying to nail down. And is that happening in our cancer patients when they get, for example, immune checkpoint therapy or chemotherapy or any other treatment? Again, from a biologic standpoint, it would make sense that there's an impact between them because they're both sensing the environment. The immune system is sensing the environment in order to have a response. Nerves are sensing the environment in order to have a response. And there has to be some crosstalk that's happening. So I don't think it's a crazy idea that to expect that we'll find the mechanisms about how these different cells are interacting and which neurotransmitters are affecting which immune response. I think that's still things that we're trying to figure out, whether it's cytokine production or trafficking or any of those things. I think those questions are still up for debate.
I think when we talk about discovery of mechanisms and pathways, it seems like we're discussing the value of human-based studies that inform mechanistic studies in the mouse and mechanistic studies in the mouse that inform human studies. And I know that the two of you have championed this reverse translational strategy. Maybe you could sort of briefly describe it and how that might be a unifying approach to building a pipeline for iterative discovery of the next generation of therapies.
Absolutely. I mean, it's an iterative process, right? Going between humans and mice. So obviously, from the immune checkpoint therapy space, we can talk about Jim in mice, right? He did the work to show that CTLA-4 was an important player in controlling T cell responses and then developed the anti-CTLA-4 antibody. But how does that affect a human immune response? We needed to really go back to actual humans and look at what the immune response is there. And that's what we did. And then we could understand some pathways that we found. For example, the ICOS pathway was one of them, interferon gamma. Other things were there in the human responses that we could then go back and test in mouse models to see, are they really relevant? Is this really a mechanistic pathway that we can consider targeting? And I think this is going back and forth. So this idea of reverse translation came about from the fact that once the field exploded, we had so many immune checkpoint therapy studies ongoing. I think if you looked in clinicaltrials.gov, we have well over 3,000 clinical trials with immune checkpoint therapy. So clearly the clinic is outstripping the laboratory science. And so if the clinic has so many patients, we need to take advantage of learning from each one of these patients. And that's the reverse translation model, focusing our attention there, getting all of the tumor samples and blood samples from these patients longitudinally over time, so we can see the dynamic changes that are happening and then formulate hypotheses that we can then take back to the lab and test mechanistically in mouse models. Bring that data back so that once we understand the mechanisms that are appropriate for resistance or a mechanism that can help improve the response, bring that back in a way in a combination trial in patients again and keep going back and forth in this way. So I think it's a very important model. We set that model up in 2004 when I first joined MD Anderson, and I'm happy to see that many, many more people are now using it.
So you mentioned a very important point, the sense of like feeling alone early on in your way, going to AACR, being at the last day of the meeting. And I remember I had the same feeling a couple of years ago, and now with with the cancer neuroscience, again, kind of exploding and having conferences like the one that we're going to have in Houston. Um, what do you guys want to tell to those like young investigators that are, really working hard to make those discoveries in a field that until like 3, 4 years ago did not exist?
Yeah.
Uh, what would you tell them to keep them on?
Well, we know they're neurotransmitters that found in tumors, and we know they're neurotransmitter receptors on molecule on components of the immune system. We need to sort that all out, like we have begun to sort out the networks in the immune system. So it's a lot to do.
I
Great.
would just, yeah, I would just say, again, the questions are so important, right? The field requires dedicated scientists, curious minds, really wonderful collaborations, and they're all welcome to keep doing that. We want people to keep doing science. I know right now it's a little bit murky about, how is science going to move forward, but I want everybody to know that, the scientists, we don't come into this with the idea that, there's going to be a major breakthrough every day, right? We know that sometimes it's a slog and you have to work through it and you have to come back to the lab bench and do it. But we do it because we're passionate about understanding the field. We're passionate about understanding and, identifying the mechanisms. And we know that there are mechanisms to be identified. And in cancer and neuroscience, there are clearly mechanisms to be identified. So those junior people working on this just have to, sort of go with the energy, go with the passion, be persistent and keep doing the work because it will lead to this field having major impact as we continue.
Yeah, I was always fascinated, Pam. I know how hard, you work in order to push all those clinical trials, and you're working with so many scientists. Where do you find your energy from? Only God knows. You're so passionate about it. It's one thing you can't infuse in people, right? You can train them almost to do anything, but this passion is, yeah. And how do you deal, early on with criticism? Like Jim and Pam, when you first, when you made the discovery with the CTLA4 and it was just you and Pam, when you said, okay, we need to like move this to clinic in bladder cancer, and probably your colleagues were a little bit skeptical. How do you deal with this as a scientist, as a physician scientist, or as a person?
Yeah. I mean, criticisms are part of, our daily life. I mean, I think it's okay. I think you have to sort of be like, okay, but this is what I want to do. I'm sure you have your supporters as well, right? So it's not just that we have our critics, we have our supporters. And so you lean into the supporters and then you try to convert the critics to be your supporters by showing them data. I always think that, for me, data speaks for itself, right? And so I try not to bombard people with just my words, but I try to show them the data and let them make a decision based on the scientific data. And it's hard to argue with a patient CT scan where the tumor goes away completely. After a while, everybody jumps on that bandwagon. And so then there are very few critics left. But you do have to walk that line in the beginning of trying to lean into your supporters, and listen to your critics. Sometimes they're giving you valuable advice, right? So don't just ignore them. Sometimes they really are telling you things that you have to pay attention to, and then you should bring them back to data later, and hopefully your critics become your supporters. But I think that's just part of normal life. And, we've all been through it in different ways. I still remember when I had 3 kids and doing my MD PhD training, and people were like, well, you can't be a mom and a clinician and a scientist. And I was like, well, just watch me. I mean, at the end of the day, you just have to do it. I mean, you're not going to make everyone happy, but that's okay. You do what makes you happy.
Sometimes it can be very frustrating, though. I mean, what I ran into is people said, well, there have been all these trials, immunotherapy trials with vaccines and stuff, and they never work. So what you're talking about won't work. And I said, you're a scientist? I don't think so. If you were, no scientist would ever say anything like that, so you should shut up, and listen to data. I mean, Pam has it exactly right. And if you have the data and you've tested your own data to the extent as far as you could go, and it holds up, then you just got to stick with it and keep going. And if you think it's important, you just got to keep at it, no matter what people say.
It's so funny. I remember like the first time I saw that those neurons are actually changing their behavior and their phenotype in the context of cancer. Someone who did some neuroscience before said like, that cannot be true. I had to repeat this experiment. I did it like I think for 6 months. I repeated it over and over again before I went to my advisor and told him, "Hey, dude, I think that's what's happening." I had really to convince myself, and from that point, it was easy because I replicated this 6 times.
Yeah. You should be on.
It kind of sounds like a primer on scientific resilience
Yeah, exactly.
and how to fight the power, if you will, which is the status quo thinking on how things work.
Yeah.
This is perhaps a good place to end. This chapter. I think the next chapter is the conference, and I just want to say thank you both for taking the time to speak with us, and we so look forward to continuing to learn about your work, to receive that support that you're giving Moran and the cancer neuroscience field more broadly, and just thank you. It's just so wonderful to have an opportunity to talk with you.
This was a lot of fun. Thank you for having us.
Sure.
Thank you so much. Thank you, Jim and Pam.
Thank you, Moran. Bye, everyone.
Bye-bye.
Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.